Okay, I think that we're ready to get started. So thank you all for joining us. My name is Manisha. I'm a program manager at Seven Bridges. I work with the Cancer Genomics Cloud, and I'm very excited to tell you today about some tools that we've developed to make it easier for single-cell researchers to do their data analysis on the Seven Bridges platforms. Today we'll be giving a quick overview of the platforms themselves, and then we'll go into the tools that we now have available on those platforms for single-cell research. Then we'll end on some case studies that go into some detail. And I'll be presenting the introduction, and my colleague Nemanja will be presenting the bulk of the talk on the actual tools themselves. So just to give you a sense of where the talk is going, we're going to start with an overview of the platforms, what they are, the capabilities they offer to researchers, and then we'll highlight some of the high-level concepts that are important for working with single-cell data. And then we will go into a deeper dive of the actual tools that we now have available on the platforms for doing single-cell research. And we'll end with some use cases and comparisons and case studies showing the actual tools themselves in use on real single-cell data. So Seven Bridges has been around for quite some time. We are a bioinformatics company, and our goal is to make it easier for researchers to do their data analysis. For the past 10 years, we've been refining how we make it easier to do that by developing these industry-leading platforms that help researchers from all ends of the scientific spectrum. So we have capabilities that are useful for basic researchers that may be doing basic bench research and have data analysis needs, all the way through to a bioinformatics scientist at a pharmaceutical company who's maybe analyzing clinical trial data. And this depth and breadth of experience with many different types of bioinformatic analyses has been worked into our platform over the years, and we've been awarded a number of awards for how easy our platforms make it to do these types of analyses. And the way we do this is sort of shown in this diagram here. And our overall goal is basically to shorten the time from hypothesis generation to insight by reducing the amount of effort that goes into the iterative cycles where you're testing and retesting your hypothesis that every researcher has to go through. And we do this by co-localizing a set of features of the platform that make it really easy to get through that iterative analysis easier by standardizing how that analysis can be done. So basically, we co-localize the access to data, whether it's public data sets or bringing in your own private data, the tools and workflows needed to do the analysis. And again, we have many publicly available tools on the platform, or we offer the flexibility to bring in any of your own tools. And combining this with the scale and flexibility and speed of cloud computing. And these three things together really give us the ability to do very high volume processing very quickly and easily. So that scalability to deal with very, very large data sets, very, very complex analyses. The entire platform is built with security in mind. So every concept of the platform is security first. So you, the scientist, don't have to worry about security, and you can collaborate with other researchers around the world very, very easily. And it's very easy to share data. It's very easy to control your data. And this makes it much easier to work together on data sets and come to a conclusion faster. We have a lot of flexibility built into the platform as well. So we have a graphical user interface that allows you to set up and run workflows very quickly and easily. But if you're more of an advanced developer, we also have an API layer that can be accessed. So you can programmatically get involved in any aspect of the analysis you wish to get involved with. And the combination of all of these things together really lets you take complex data sets and get to a conclusion much more quickly. And that can lead to things like drugs being developed more quickly. So looking at complex data and understanding which drugs make sense to move forward with, or a complex single cell data set and understanding which cell types are the key ones to move forward with. And that's really how we use the capabilities of our platforms to make it easier for scientists to go from their hypothesis all the way through to their conclusion and make the science happen more quickly and easily. The platforms are all built with FAIR principles in mind, which is findable, accessible, interoperable, and reproducible. And we do this by engaging with many of the community standards organizations, such as the GA4GH. So by engaging with these communities, we build these standards and principles into the platform, and we've incorporated many of these standards. So again, reproducibility, workflow portability, security, and sharing data have all been baked into the platform. So you can be sure that the data you're doing will be easily shareable if you wish to share it, and secure if you don't wish to share it, and that the tools and workflows you're using are portable and reproducible, and the results can be reproduced by anyone around the world. So by engaging with these standards bodies, we are able to provide a system that's extremely reproducible and interoperable. We have several platforms that address different research needs. The Seven Bridges platform is designed for the needs of biotechs and biopharma companies. So this is primarily designed to answer questions related to drug discovery and clinical trials. We also have a series of platforms that are government funded and designed for academic or nonprofit use. And these have been designed to meet the needs of different research communities. So the Cancer Genomics Cloud is an NCI funded platform that primarily works with cancer researchers. And the data we're going to show you today and the work we're presenting today was primarily done on the Cancer Genomics Cloud. Cavatica is our platform for working with pediatric diseases. And Biodata Catalyst is a new platform for working with cardiovascular diseases. So the need for these types of cloud computing in genomics and other areas of science has really been driven by the expansion of data sets. The Cancer Genome Atlas is one of the first data sets that really made it clear that the benefits of cloud computing would offer to the research community, because the TCGA data set is huge. It's petabytes of data, and it became increasingly difficult for a researcher to access that data. So downloading it was extremely time consuming. And then once you had downloaded these huge files, doing an analysis on such complex data sets would take a huge amount of computing power. By moving that data to the cloud and taking advantage of cloud platforms, researchers no longer have to worry about this. They can access the data from anywhere with an internet connection and be able to, and anyone can analyze these huge data sets now quickly and easily without having to be experts in computational infrastructure themselves. We've seen a similar trend with the single cell fields. So 10 years ago, analyzing one single cell was a big challenge. But as the technology has evolved, the ability to do millions and millions of single cells in a single experiment is now kind of the standard. And you can imagine these data sets are huge, where every individual cell could be an entire transcriptome or entire genome. The size and complexity of these data sets makes it really difficult for users to access them, to share them, to perform analyses that are really computational intensive on these huge data sets. So again, this is where cloud computing can really democratize access to doing this type of research by keeping the data in the cloud where anyone can access it. And anyone can now take advantage of these types of data and these types of tools to do single cell research. We provide access to many different types of public data sets. So one of the three pillars, access to data, tools, and computational power. We have a whole bunch of different types of data that we also provide access to. While many of our data sets are genomics, we also have some proteomics and imaging and other types of clinical data. And the data that we're going to talk about today is from the Human Cell Atlas data set, which is all single cell data. We also provide a lot of ways to use different tools. So we have a public apps gallery that has over 400 different tools and workflows that allow you to put together any workflow without necessarily being expert in that particular bioinformatics method. And we cover many, many areas of analysis in our public apps gallery. All the tools on our platform are wrapped in common workflow language and use Docker containers, so they're very portable and very reproducible. If you want to edit one of our public tools or bring in your own tools, we have a really nice piece of software to do this called Rabix Composer, which is shown in the animation on the right here. What Rabix Composer allows you to do is graphically build tools. You can set the inputs and outputs merely by dragging and dropping, as you can see here. and all the other parameters. And when you're finished, everything will be neatly wrapped in common workflow language and available to run on the platform. So we make it really, really easy to bring in your own tools. And it's also very flexible as well. So you can, every version of the tool you make will be saved. Every parameter you can choose to either expose to other users or fix so it cannot be changed. And all of these parameters are saved every time you run the tool. So running a tool today, you should get the exact same results as if a collaborator runs it a year from now. So everything is extremely reproducible. And we also have a set of tools called for running Jupyter and R packages in the cloud as well. So not only do we have the tools to do primary data processing with our tools and workflows, we also have integrations that allow you to run notebooks and our studio packages in the cloud as well. So without having to download the data, you can take the results of your analyses and then go on and do more complicated higher level analyses or visualizations directly integrated with your same project in the cloud. And this offers the same benefits of being able to collaboratively work on projects together in a secure way. And many of the tools that we're going to talk about today are R packages for single cell analysis. So Nemanja will go into more detail about some of the R packages we now have available on the Seven Bridges platforms for doing this type of analysis. So now I'm going to switch gears a bit and talk about why we do single cell analysis and how the platforms help us do it. So the reason why single cell analysis has grown so much in the past several years and been so impactful in transforming many different fields of biology is that understanding the individual pieces that go into a tissue or a system is hugely important to understanding the properties that emerge from that tissue. So looking at, for example, cancer stem cells can give you a much better sense of how cancer progresses. And by looking at things individually, you can get greater insights from the smaller populations that are likely driving the true biological changes, like, again, for example, cancer stem cells. And you can also use these insights to identify potential marker genes for therapeutic targets or biomarkers. However, as the pace of single cell research has exploded, there continue to be lots of challenges with them. One of the primary challenges is, as I mentioned earlier, the data sets can be huge and very complex. So millions of data points, each of which is a whole transcriptome, a whole genome, et cetera. And this can make it really difficult to access and compute on these data sets. And that's where the Seven Bridges can really help a lot by keeping the data in the cloud and the analyses in the cloud. We make it really easy for anybody to be able to access and compute on these large data sets. And then while these data sets are very large and complex, the actual individual data from a single cell is not that huge. So they're actually, the data itself is actually quite sparse compared to a bulk RNA-seq experiment, for example. So this means new methods for looking at this type of data have to be developed. And in the past 5 to 10 years, there's been a huge number of tools developed. You can see on the graph to the right from an organization that keeps track of these tools, there's over 600 tools now available for doing single cell analysis. And there hasn't quite been a consensus yet in the field on which tools to use. So it can be quite confusing for somebody new to the field to know exactly which tool to use and how to set it up. And this is, again, where the platform can be very helpful. We have publicly accessible apps, so you don't have to build the tool yourself. They're pre-built on the platform for you. And what we're going to do today, actually, is talk through some of the most popular tools that we've added to the platform and do some comparisons between them to make it a little bit more clear which tool might be most helpful for your research needs. And you can see the growth in the field by the growth in single cell atlases that have come out in the past few years. So the first set of atlases that came out, not surprisingly, were from very simple organisms like C. elegans and fruit flies. But more recently, more complex mammalian atlases have come out. There are multiple ones now of the entire mouse brain, the whole mouse nervous system. There are atlases out for every tissue within mouse. And there are many human projects that are in progress, which are extremely complex. So the Human Cell Atlas is aiming to characterize every healthy tissue in the body. And the Human Tumor Atlas Network, for example, is looking at single cell characterization of tumors. And you can see an example of one of these on the right here. It's looking at zebrafish developmental biology, and they were able to basically map at a single cell level every cell in the organism and identify which cell types were present in there. And they did that using the Seurat package, which we will talk about more in the rest of the talk. And not only that, they were able to look at lineage by reconstructing the developmental trajectories. So not only do they know what cell types are present, but they know how they're connected and in what order they developed by making these single cell maps. So this gives you a much more comprehensive picture of what's happening in the biological development of these organisms. And we've also made tools to do these trajectory analyses available on the platform as well. And we'll actually talk about this in the next section as well. So now I'm going to transition over to Nemanja, who will go through the actual tools themselves.
Thank you, Manisha. We can start off with the next slide, please. Okay, so today we will be talking about tools and workflows optimized for processing both UMI-based and full-length single-cell protocols. We will be presenting our packages that could be used for downstream single-cell analysis performed on gene-to-cell counts. Also, we will present the results of comparative benchmarking of different tools for processing 10x Chromium datasets in cloud-based environment. And we will talk about how we analyzed full-length data to understand changes in endothelial tumor cells over time. Next slide, please. Okay, we'll start off with presenting tools and workflows optimized for processing both UMI-based and full-length single-cell protocols. Next slide. One feature of single-cell analysis is the existence of a large number of sequencing protocols. The main difference between protocols is whether they capture the full length or just one end of a transcript. Full-length protocols developed for single cells are quite similar to protocols for bulk RNA-seq. They have lower multiplexability, meaning that you can combine fewer cells per sequencing run. The number of cells that can be sequenced highly depends on the desired sequencing depth. The full-length protocols are useful for cases where higher sequencing depth is required, such as identification of single nucleotide variants, mutations, RNA splice variants, or T-cell receptor sequencing, which is the example shown on the right. On the other hand, protocols that sequence just the ends of transcripts have introduced barcode and UMI labeling to one end of the transcript. Cell barcodes enable labeling of each cell and expands multiplexing ability to more than 1,000 cells per sequencing run. The use of UMI sequences will be explained later. Combining more cells means that the sequencing depth of these protocols are usually lower in comparison with full-length protocols, and they are used for dissecting heterogeneity of complex tissues and identification of marker genes, which could lead to detection of novel cell types. Next slide, please. So tools for processing full-length single-cell protocols are pretty much the same ones used for processing bulk RNA-seq, but optimized for single-cell data. Data produced with full-length protocols may come already demultiplexed, where each pair of FASTQ files carries raw sequencing reads of a single cell. If that is not the case, demultiplication is the required step in the analysis. Some of the splice-aware aligners, such as STAR or HiC2, can be used for aligning reads to the reference, and tools like RSAM or HT-seq can be used for quantification, producing tables of read counts per gene or per transcript as a result. There are many extensions of tools used for bulk RNA-seq that are optimized for processing single-cell data produced with UMI-based protocols, and we will be mentioning some of these in more detail later on, but the main addition to these tools are the barcode demultiplexing and UMI deduplication steps that are mandatory for UMI protocols. Next slide, please. Okay, so cell barcodes refer to the unique sequences added to each fragment that came from a single cell before all cells are mixed together and sequenced. UMI-based protocols use additional type of barcodes called UMIs. These are unique molecular identifiers that are short random sequences added to the transcript during reverse transcription. They enable sequencing reads to be assigned to individual transcript molecules and thus enable the removal of amplification noise and biases from single-cell data, which provides a great improvement in quantification step of the analysis. UMI sequencing typically produces paired-end reads, where one read from each pair captures the cell barcode and UMI, while the other read consists of exonic sequence of the transcript. Next slide, please.
Okay.
Okay, so cell barcode demultiplexing refers to the step in processing where the cell barcode information is used in order to know which sequences came from which cells after they had all been sequenced together. So the sequences with the same barcodes are grouped together. The UMI deduplication step removes the duplicate read sequences that came from the same transcript. One of the main challenges with analyzing this type of data is dealing with sequencing errors present in barcode and UMI sequences, and different single-cell tools for processing this kind of data have different approaches that can be used for processing UMI-based single-cell protocols. And tools are separated into two groups based on alignment approach they use. So on the left are the alignment-based tools such as CellRanger, Zoomis, and StarSolo that use splice-aware algorithms for aligning read to the reference that is already implemented in a star aligner. On the other end, there are extensions of Callisto and Salmon, and these are the tools that use pseudo-alignment algorithms for single-step alignment and quantification that is optimized for single-cell data. In the middle are UMI-based single-cell protocols that are supported by each of these tools. Tools are connected to the protocols they support with lines, and all of the tools support the 10x chromium protocol, and some of the tools such as Zoomis, StarSolo, and Salmon have option to arbitrarily set the start and end positions of barcode and UMI sequences in second read, thus enabling them to process data produced with practically every single-cell UMI protocol. Next slide, please.
Okay.
Okay, so we use a common workflow language to describe software packages for processing single-cell data and make them available on Seven Bridges cloud environment in a form of tools, workflows, or as interactive analysis. Tools would represent a single method for one step of analysis, while a series of tools connected together into a pipeline represent workflow. Interactive analysis on Data Cruncher are Jupyter or R Markdown notebooks, providing higher level analysis of processed data that includes visualizations and other interactive components. Next slide, please.
Okay.
So to summarize the first part of our talk, here is an overview of all single-cell tools we have available for both full-length processing, UMI-based processing, and higher-level analysis using our packages. Again, the goal of the processing step is to go from raw sequencing reads to some form of counts data. And for processing full-length single-cell data, we have a SmartSeq2 workflow, which we will describe in more details in the last part of the presentation, and two tools, Tracer and MixCR, that can be used for the reconstruction of T and B cell receptors. For processing UMI-based single-cell data, we created a Callisto bus tools workflow, We also have a cell ranger toolkit consisting of 5 separate tools, and we have extensions of star aligner that is called star solo, and salmon pseudo aligner which had extension called olivine. In the data cruncher section, which we will discuss more in the second part of the talk, we have a single cell interactive analysis, and this analysis is executed within RStudio environment of Seven Bridges Platform. And it includes notebooks that, as an input, takes count data and perform additional analysis and visualizations. We have 3 separate analyses created as our Markdown notebooks, which can be used to perform correction of patch effect using Harmony R package, clustering and detection of marker genes that is based on Seurat package, and trajectory inference based on Monocle. All of these notebooks use the results that are produced with tools and workflows that we already mentioned. Next slide, please.
As we get into the actual tool usage, I just want to mention that if you have questions, please type them into the chat window and we'll try to address as many as we can at the end.
Okay. So in the following section, we will be talking more about analysis available on Data Cruncher that can be used to perform downstream processing of gene-to-cell counts. Next slide, please. So single-cell downstream analysis start with a gene-to-cell count table produced as a result of quantification using the tools described in the previous section. And these tables hold information about the number of reads or UMIs that are assigned to each gene and each cell. Rows of the table representing names of genes, while columns representing different cells. We'll give a brief overview of some downstream analysis that could be performed on gene-to-cell counts. Next slide, please. Okay, we'll start off with the correction of patch effect. So patch effect is usually present when merging single-cell data generated at different times or different laboratories, which can introduce technical variability. So the purpose of patch correction analysis is to remove the technical variability and preserve biological variability in dataset. The best display of patch effect is visual representation of datasets reduced to 2 dimensions using TSNE. And in the top right image, we compare cells from 2 datasets, one colored blue and the other one colored orange. So the blue and orange are clearly separated on the top picture, and this is an indication of patch effect present after merging 2 datasets. After identification of the patch effect, we can remove it using the Harmony R package. On the bottom picture, which is the same data processed with the Harmony package, we observe no clear separation between cells originating from different datasets, thus confirming we have successfully removed the patch effect. Next slide, please. So the second analysis we would like to highlight is clustering and detection of marker genes that is based on Seurat R package. Seurat analysis consists of several steps, which include selection and filtration of cells based on some QC metrics, data normalization and scaling, detection of highly variable genes, dimensionality reduction using PCI, clustering of cells, visual representation of cell clusters in 2 dimensions, and detection of marker genes within cell clusters. So on the image to the right is a TSNE plot showing the results of clustering in 2 dimensions, and we observe 8 clusters representing distinctive cell types. And here cell types were detected and annotated by analyzing marker genes that are expressed in each of these clusters. Next slide, please.
Okay.
So the third package I would like to highlight is Monocle. And cells that exist in different stages of development within the same population can display different transcription profiles over some time period. The package Monocle provides methods for ordering single cells and placing them along a trajectory that corresponds to a biological process such as cell differentiation. In the first step of this analysis, it is important to remove empty cells and doublets, and those would be the places where two or more cells are sequenced by accident. And Monocle then employs a differential expression test to reduce the number of genes. This step requires prior knowledge about cell types and genes expressed in those cell types. The alternative is to perform unsupervised clustering before this step. In the next step, Monocle performs dimensionality reduction, followed by construction of minimum spanning tree. And in the final steps, Monocle algorithm finds the longest path through the minimum spanning tree, corresponding to the longest sequence of transcriptionally similar cells. We'll be talking about trajectory inference more in the final section of the presentation. Next slide, please. Okay, so now that we have gone through the tools and packages available for single-cell analysis, we will present some examples and demonstrations of how they perform. And first, we'll present the results of comparative benchmarking of different tools for processing 10x Chromium datasets. Next slide, please. Okay, so we did performance benchmarking to compare the execution runtime and AWS costs of 4 single-cell processing tools on 10x genomics datasets. So left plot represents the relation between total size of 10x dataset and execution time on Seven Bridges Platform. And on the right plot, you can see the relation between the dataset size and AWS costs. We observed that pseudo-alignment based tools such as Salmon and Callisto, and that would be the ones in the plot that are colored in blue and red, and those have significantly better performance in terms of execution time and AWS cost in comparison to alignment-based tools such as CellRanger and Zoomis. On the following slides, we will talk more about methods we use to compare the precision of the obtained results. So we compare the accuracy of the results produced with different tools on multiple levels of single-cell analysis. For the quantification level comparison, we simulated a single-cell dataset and also used a bulk RNA-seq data as a second approach. We also performed clustering and gene marker comparisons using interactive analysis based on Seurat package. And for this purpose, we use two datasets that are labeled as gold standard and silver standard. And this gold standard dataset is a low complexity dataset created as a mixture of 3 human cell lines that are mixed in equal proportions. And the silver standard dataset is a real complexity dataset. It is representing peripheral blood mononuclear cells, and this dataset is downloaded from 10x Genomics website. So cell type labels for each cell and marker genes for distinctive cell types were available, and we used them as a ground truth in our comparisons. And since the ground truth information related to this dataset is obtained using a different approach that is less reliable than used for gold standard dataset, this one is labeled as a silver standard. So we'll present the results for the silver standard dataset because they are more divergent and provide better insight when estimating precision of these tools. Next slide, please.
Okay.
So we processed simulated FASTQ files with 4 different tools and used gene-to-cell counts as a ground truth for comparing precision of quantification results. Prior to comparisons, genes were split in 10 groups based on the sequence uniqueness. Salmon, Callisto, and CellRanger tools showed high correlations to the truth datasets for the highly unique genes, while Callisto had the highest correlation in the groups of moderately unique genes. On the other hand, Zoomis showed the lowest correlation to the truth dataset in comparison to other tools, and based on these results, we can say that Callisto on this test performs best. Next slide, please.
Okay.
So here we compare the results of clustering performed on silver standard dataset, and we used adjusted rand index and hit homogeneity score for validating the results of clustering given ground truth. And both scores will have a value close to 0 for random labeling and exactly 1 when the cluster labels are identical to truth. So here we purposely enforced the identification of 11 cell clusters because this many reference subpopulations was reported in truth dataset. Also, we performed analysis with Seurat's default settings. From the table, we observe Salmon and Callisto had slightly better adjusted rand index score and homogeneity score than the Cell Ranger, while Zoomies had the lowest scores in comparison to other tools. Again, Callisto seems to be performing the best in comparison to others. Next slide, please.
Okay.
Here we use Seurat to identify marker genes expressed in distinctive cell clusters in a silver standard dataset. And based on identification of marker genes, all of the tools were able to identify the majority of cell types. But again, Callisto performed best, enabling detection of 8 out of 11 different cell populations. On table below, you can see that plus sign means that cell type was identified. And we observed that no tool was able to identify all cell types that exist in dataset. But nevertheless, Callisto was able to identify one more than the others. Okay, next slide, please.
Okay.
So our benchmarking results indicate that pseudo-alignment based tools are much faster and require far less computational power than tools based on star aligner when processing 10x single cell datasets. Pseudo-alignment based tools give accurate results and in some segments outperform star-based tools, which we have witnessed on multiple steps when making comparisons. But nevertheless, we advise that Cell Ranger software be used on 10x data since it is optimized and extensively tested and validated. Additionally, Cell Ranger software provides ability to perform clustering and detection of marker genes without the use of downstream analysis based on Seurat. And the final remark would be to use pseudo-aligners rather than Zoomies as an alternative to Cell Ranger when working with 10x data. Okay, next slide, please.
Okay.
This would be our last example of how these tools can be used, and we would like to present the trajectory inference analysis conducted on full-length data from endothelial tumor cells that are isolated from mice with melanoma. Next slide, please.
Okay.
So to demonstrate performance of the tools for processing full-length protocols and packages for trajectory inference, we perform some additional analysis using a dataset that is available on the Human Cell Atlas data portal. And this dataset comes from a study on tumor development in mice that is conducted in Teichmann laboratory. And the authors performed single-cell RNA sequencing to analyze changes in stromal cells, such as endothelial cells, fibroblasts, and immune cells at different stages of tumor development. They were tracking these changes in mice to whom they injected melanoma cells, and they were collecting samples after 5, 8, and 11 days after injection. And the main focus of the analysis was related to changes in development of T cells and fibroblasts. Therefore, our idea was to expand this analysis to additional cell types by investigating changes in endothelial tumor cells. Next slide, please.
Okay.
So the authors used SmartSeq2 full-length protocol to produce raw sequencing reads. Reads in FASTQ format are available on data portal of Human Cell Atlas, which we recently made available on Cancer Genomics Cloud platform. And to process files, FASTQ files, we created an implementation of SmartSeq2 workflow in common workflow language, and we optimize it to be executed on Seven Bridges cloud infrastructure. The original pipeline is created by the data coordination platform of Human Cell Atlas, and it uses HiSet2 for the alignment and RSEM tool for quantification. And the entire dataset contains more than 12,000 paired and FASTQ files, but using the metadata associated with files, we were able to select just the files representing endothelial tumor cells. We processed this subset of endothelial tumor cells, which had more than 1,000 files, paired and FASTQ files, and total processing time on Cancer Genomics Cloud platform was 49 minutes. The analysis was running on 10 parallel AWS instances, each having 36 CPUs and 72 gigabytes of random access memory with total price of $5.5. Next slide, please.
Okay.
After processing raw sequencing reads and producing gene-to-cell counts, we performed trajectory inference analysis using two different approaches. So both of these approaches are using Monocle R package, but the first approach incorporates timescale information where we identify differentially expressed genes between cells from different stages of tumor development. So before composing the trajectory, we filter out empty cells and doublets. We start analysis with Monocle by selecting the genes that are differentially expressed between different times in development. We use these genes in the later steps to perform dimensionality reduction, followed by the construction of a minimum spanning tree. In the final step of the analysis, the Monocle algorithm maps each cell to the trajectory and finds the longest path through the minimum spanning tree, which is the line connecting the largest number of cells. On the top right plot, we show a trajectory along which Monocle placed individual cells, and selecting differentially expressed genes at the beginning of the analysis, we enforce this trajectory to be in alignment with changes in endothelial cells over time. Now that we calculated the trajectory, the additional step in the analysis could be identification of genes that change along this trajectory. On the bottom right plot, you can see 3 genes coding for transcriptional cofactors and changes in their activity over time. You may notice that these genes were not active in early stages of tumor development, but their activity increased in later stages. So this approach could potentially be used for the identification of genes as a potential biomarkers indicating progression of tumor through different stages of development. Next slide, please. And here we would like to demonstrate another, a more comprehensive method when analyzing trajectories of tumors. So in the second approach, instead of selecting differentially expressed genes over time, we use all genes that pass some filtering thresholds. And we use those genes to perform clustering. Then we order the cells based on the results of clustering, and the later steps in constructing the trajectory are the same as in previous example. As a result, you can see that on the top right plot, the constructed trajectory has more branching points, and the cells from different stages of development are more uniformly distributed along the trajectory. So This approach more realistically represents the development process in tumor tissue, where you will have one trajectory along which the majority of cells are aligned, but also many branches which could represent cells that carry some clonal mutations. On the bottom plot, you can see that Monocle splits this trajectory into states. So the additional analysis could be the identification of marker genes for each state that could represent a potential clonal expansion. Additionally, you could use the information about times when cells were collected to specifically track down cells that are at the origin of clonal expansions. So thus this second approach could provide a significant information about cells that are drivers of somatic mutation in tumor development and use it to create therapy that will most effectively target tumor tissue. Okay, next slide, please. So I would like to finish this presentation with a couple of ideas for application of single-cell sequencing and how the Seven Bridges platform could be facilitated for the future clinical trials. So we mentioned the Human Cell Atlas project that aims to characterize every cell type with the focus on healthy individuals. And this information is crucial for understanding how disease states are different from healthy tissues. One example would be recent finding that the majority of CFTR transcripts in lungs are coming from a novel cell type that was not identified before. So CFTR gene, when mutated, is the main cause of a disease called cystic fibrosis. But now knowing which cell populations are most involved in producing these disease-related transcripts could help scientists create targeted therapy approaches. Another application of single-cell sequencing would be to monitor disease progression and response to therapy, which is highly utilized in the area of cancer research. You can perform trajectory inference to track down which clones or cells are eliminated by chemotherapy and which ones persist or become resistant over time. Also, single-cell transcriptomics gives potential for mapping patients to the right cancer treatment. And additionally, how well a drug works also depends on what is happening in the surrounding tissue. Single-cell analysis of tumor environments provides a complete picture of disease progression and patient's response to treatment. And we showed a segment of such analysis in our presentation today. So all of these and many other questions can be answered using single-cell transcriptomics in combination with bioinformatic tools we presented today, and cloud-oriented infrastructures like Seven Bridges Platform could help manage increasing sizes of single-cell data sets and provide the environment to conduct reproducible bioinformatic analysis. Okay. Thank you all for listening. So that will be all for my side. So, Manisha, would you take over if you have any questions?
Yeah, so we have a few minutes left for questions. Thank you so much for that really helpful presentation, Nemanja. If you do have questions, please type them into the chat window in the webinar. One question that we've gotten is whether this webinar was recorded, and indeed it was recorded. So we'd be happy to take any questions now. So maybe one question, why do you think that the Zoomies tool performed so much worse than the other tools in the benchmarking analysis?
So the question is why. So both Zoomies and Cell Ranger are using the star aligner in the background, but I suppose the key thing here is the quantification step that is obviously a better optimized in the Cell Ranger, and also in the tools that are using pseudo-alignment based algorithms than it is in the zoomies.
What are sort of the benefits and trade-offs of the regular aligners versus the pseudo aligners?
Okay, so in general, regular splice-aware aligners are used to map reads to the mostly genome reference. So you are able to identify potentially new isoforms, and they are more precise when identifying, for example, mutations, SNVs, and so on. But on the other hand, pseudo-aligners are much faster, require far less computational power, and they usually use transcriptome reference for the alignment step, thus not being able to detect novel isoforms. But I think the Salmon, in one of the latest releases, also introduced the combination of genome and transcriptome reference in order to overcome these limitations. So yeah, I think the quantification of the reads is whether gene expression estimation is quite good as in aligners, but they could not be used for things like identification of novel isoforms or mutations, splice variants, and so on.
Are there other tools that you've considered adding or will be added soon to the platforms in addition to these?
Not at the moment, because I think we covered a large number of protocols that can be processed. What we are missing here is in the benchmarking, we did not include star solo, and that would be interesting to compare this tool to other tools based on star and to compare against the results from pseudo-aligners.
Are there tools available for single-cell CNV analysis from single-cell sequencing from the 10x platform?
So the current focus of our study was single-cell transcriptomics. But like I said, we have the Cell Ranger toolkit for processing 10x dataset. I'm not aware if any of these tools are compatible for CNV identification. That should be checked. But yeah, we have a Cell Ranger software that could potentially analyze this type of data.
Yeah, and generally there's far fewer tools on CNV analysis for single cell data than RNA-seq, but it's something that's evolving. Did you use the latest version of Monocle? And are there other versions you should consider?
Yeah, so at the time when we were doing this analysis, the latest version was still in Monocle 3 was in alpha version, so we used Monocle 2. And after that I tried out Monocle 3, but it doesn't provide some of the options that we presented today. So I think if you have like if you would like more customizable analysis, trajectory analysis, and where you'll be able to apply different approaches, that the Monocle 2 is better than Monocle 3. But yeah, I think that Monocle 3 has just been released, and we should definitely compare some of the results between those two.
Couple quick questions. The benchmarking we presented was on the 3' end counting data, not on the full length data. to clarify that. And we got a question about which tools are available on which platforms. We're aiming to get all the tools available on all platforms soon. Here's an interesting question. Recent paper found that comparing STAR, Callisto, and BWA pipelines, that Callisto had more dropouts compared to STAR. Have we seen anything like that in our data?
Yeah, we actually did not like do that sort of comparison, but yeah, I'm not sure about which dropout rates are referring this.
Yeah, maybe we can follow up on that. The link to the paper is here, so maybe we can take a look and follow up. Yeah. And I guess maybe one more quick question. Where did you obtain the annotation information for the different cell types, like the CD8 cells?
So we used publicly available datasets that are downloaded from 10x, and that would be peripheral blood mononuclear cells. And these cells are well characterized. And I think that we obtained these annotations from a publication that came from a research group from 10x. But yeah, I think that for these data sets, yeah, these annotations are pretty much available everywhere.
Okay, so I think that's all the time we have today. Thank you so much for your participation and all the great questions. We will be making this available after the fact, and I hope this was really helpful and encourages you to continue on with your single cell research. So thank you so much.